Okay, so pretty sure most of you are kind of familiar with the concept of precision oncology. So the idea is that you want to learn as much as possible about the tumor of the patient to see what makes it really unique. A lot of patients present themselves to the clinic with the same kind of clinical variables, same age, same cancer type, same sex, and despite those facts, they actually respond differently to a given therapy. So we believe that by studying tumor very precisely and their microenvironment, we'll be able to kind of learn vulnerabilities that are very specific to a given tumor. And to do so, you have a lot of different technologies you can play with. You can interrogate the tumor at the genomics level, looking at mutation, copy number variation, etc. At the epigenomics level, transcriptomics, proteomics, metabolomics. You can even grow those tumors into the lab and challenge them with drugs to actually learn what kills them. So very, very rich multimodal data you can use, or multimodal technologies you can use to study the tumor. And so, question for you, if you had to invest in one technology to find a way to predict how a tumor is going to respond to drug, what would that be? And in a way, you probably know that DNA is the one we do currently in the clinic. We're interested. Okay. So a lot of people are fan of proteins, and indeed, proteins are like the actor in the system. Like if you can measure proteins, you have a lot of information about how tumor may respond to a pharmacological perturbation. The problem with proteins is that it's very, very hard to measure. If you use mass spectrometry or PPA, you can have a hard time to measure a lot of proteins in a very quantitative way. Actually, I'm a big fan of chromatin accessibility. I think it's kind of the sweet spot between DNA, which is very stable, but not really information rich, and RNA and proteins, which are extremely rich information rich, but are kind of noisy. Like if you do it a day apart, or if you change the temperature in the room, or the way you collect the samples, RNA and protein may change a lot. But chromatin accessibility is kind of pretty stable. It's richer than DNA. Okay, thank you very much for voting. So a biomarker is anything you can measure, right? So some biomarkers are kind of predictive of a diagnostic or a disease state, like a cancer subtype, for instance. What kind of cancer do you have? Or if we don't do anything to that tumor, what's going to be natural evolution of the tumor? So that's the prognostic aspect, like trying to predict patient survival. Today, we're going to focus on predictive biomarkers. You try to predict the evolution of the tumor conditional to a pharmacological perturbation, so under a drug response. And you can measure them using, as we saw, very, very different technologies. Some are very cheap, like staining and microarrays. Some are a bit more expensive, like RNA-seq. You can do very simple biomarkers, just one gene mutation or one gene fusion. But you can have way more complex multivariable biomarkers. And there are some of those biomarkers like that, or sometimes signatures or predictors that are used in the clinic. And you can use either a single modality, just RNA or just DNA, or you can try to combine multiple modalities. And so if you wish, before we could sequence the tumor, we could not really match patients to trials based on those vulnerabilities. We didn't really have access to the data. So we had no kind of actionable mutation or actionable vulnerability. But since we've done more and more sequencing in the clinic, we can start using that information to kind of direct patients to trials where they're more likely to benefit. And a lot of trials now have genomic aberrations as part of their eligibility criteria. So if you need the EGFR mutation to be on that trial, for instance. And so people have done more and more of those trials. And sometimes you find a mutation that you know is important, an aberration you know is important, it's associated with a given drug, but you don't really have an active trial investigating that drug in your institution. So you could match the patient, but there is nowhere to match it. And so if you look at multiple trials, it's kind of an old review, but still relevant. If you look at all those trials worldwide that are kind of biomarker driven, you can see that there is a bit of an issue here. There's a lot of red. There's a lot of patients that could not be matched based on the sequencing results. And so basically, despite very, very large investment in precision oncology, in sequencing tumors with those small sequencing assays, let's on average, 50% of the patients don't really even benefit from that approach. And that could be due to multiple challenges. We already discussed access to trials. Maybe in your institution, the right trial is not ongoing. sometimes don't have access to drugs, so it's not subsidized, not reimbursed, so they cannot really provide that as an option. The sequencing panels that we use in the clinic are really small, usually 50 to 500 genes. And if you look at the 26,000 protein coding genes, or even the 200,000 potential isoforms, it's really, really limited, but it's very precise. And most of the time, we only sequence at one level, DNA or RNA, currently in the clinic. But I would argue probably the biggest bottleneck is that we have very little of those non-gene drug associations. So sometimes we see aberration. Let's say you have aberration is the gene in the gene SLC21. You just don't know what it means. Is it going to be predictive to response to drug X? You just don't know, right? So you see aberration that could be even driver mutations, drive the tumor progression, but you don't really have any therapy to target it, or you don't know if it's a biomarker. And if you look at OncoKP, which is a FDA-approved resources for those biomarkers, we only have 79 genes involved as biomarkers, as predictive biomarkers, and that's only for 135 drugs. And we have around 1,200 drugs now that we use in the clinic, or at least we investigate in the clinic. So you can see that those biomarkers are just really the top of the iceberg. We should do better. So can we build new predictors of drug response? And so here's a very kind of naive pipeline of, I put AI because AI is fancy, but it's machine learning, biostat, name it. So basically you start with some clinical information like the previous talk and a biopsy. You can do the profiling, but you could also use radiology or pathology, but here we're focusing on genomics mostly. You have to process those data. There is nothing simple about that. We'll talk about that a little bit. Then you do the predictive modeling, and then ideally you have to do very, very robust performance evaluation. Some retrospective, like the previous talk. And if you're lucky enough and you believe really that you have strong evidence for biomarkers, then you go prospective. You try to implement that in the trial or in the clinic and see what happens. It's hard to predict, especially the future. That's why you need prospective evaluation. You have to start by asking yourself, what kind of materials do I want to use to identify my biomarker or biomarkers? Obviously, if you want to help the patients, I think patient material is the best. The problem is that if you really want a large clinical cohort, it's going to cost you a lot of money. And you can only look at drugs that have been approved for clinical use, obviously. So very high cost, very lengthy, but it's very, very clinically relevant. You kind of mimic the kind of samples you're going to see in the clinic. If you go on the lab side, then you can start cloning that tumor and treat that tumor with crazy drugs, right? Especially in vitro, you have no ethical limitations per se. Mouse models, you have some ethical considerations, but at the end of the day, you can clone that tumor and treat that tumor with many, many drugs. So you can generate a lot more data than you can do in the clinic. We always kind of, maybe some of you are wet labs and some are not, but a lot of you will probably be exposed with data from cell lines. We use them extensively in the lab to generate pharmacogenomics data. But we always say they're bad, but we always use them. But they're bad, but we use them, right? So do you think that we should even use them for biomarker discovery? Nobody's in love. Some are fairly happy with them. I gave the same talk 2 days ago. I was that fast. No, I'm that fast. I don't know. I'm in good mood. But bottom line is that they could be dangerous to use. Like sometimes you can really be misled by using the wrong cell line, or they completely drifted, right? They don't look anything like real tumor. But most of the time they're actually informative. So I think you kind of get it right. It's like yes, but. So if you look at most clinically approved biomarkers, they're very simple, the univariable biomarkers, and they have been discovered in cell lines. So we cannot say cell lines are crap. They have been useful. But no, we kind of scratch it. We've done the top of the iceberg. Now we have to go deeper, and maybe some of them are not that relevant anymore. But you have a lot of them. You can buy basically more than 1,500 cancer cell lines nowadays. And they're very cheap to use. They're easy to grow. You can use big robots, generate tons and tons of data. So definitely a lot of potential for like repetition and large data sets. And my argument is that in a way, what makes them bad also is a strength. They're very, very simple model. You control their growth media. They grow in 2D. So in a way, if you develop a new machine learning method that's supposed to kind of find biomarkers in cell lines, if it doesn't work in cell lines, which is a very simple system. Why would it work in the patient where it's a much more complex system? So in a way, from a machine learning perspective, it's a great sandbox to make sure that your method kind of go beyond technical and biological noise, in a fairly simple model. And so what a lot of people do, they take those cancer cell lines, then they go to kind of higher complexity organisms, not organisms, preclinical models, such as organoids growing in 3D, or mouse models where you engraft the tumor. And if that works, they go to the patient and they try. Obviously, there might be some false negative there. You may dismiss some important biomarkers, and sometimes they are false positive. Even if it works preclinically, it's not going to work in the patient for many reasons. But at the very least, this is kind of a path that pharma companies and researchers alike have used quite a bit. And so, what kind of pharmaco, even radiogenomic data have been generated so far? So the way you do that is that you kind of grow your cancer cells, and you have kind of infinite amount of materials. To be honest, you can let them grow, do all the sequencing you want. In parallel, and that's very the thing that's a little bit unique to biomarker discovery, is that you can challenge them with perturbation. It could be radiation, it could be pharmacology, it could be even genetic perturbation. You can do CRISPR and see what happens. And then you kind of measure their fitness, whether it's cell viability or tumor growth over time, volume, gene ancestry. You have many, many ways you can record the outcome. And so basically you have this very complex input feature, all your molecular features. Then you have this somewhat complex phenotype. It's a curve that we like to summarize in a number just to make your life easier. But the reality is that there are many different ways the tumor behaves under perturbation. And then what you want to do is to basically find this formula f of x that can predict from x what's gonna happen to the cancer cells. And it is not general AI. It's very, very narrow. You give it one task. It's probably not gonna take over the world. What I really like about those preclinical models is that people didn't feel that they should keep those data secret. Actually, you can buy those cell lines. So people kind of felt free to share a lot of that data publicly. It started with the NCI in the 90s. And then you can see over time, even pharmaceutical companies, big research groups, kind of started to screen more and more cell lines with more and more drugs. So it's really nice to see that people kept sharing over time. And so you have a plethora of data to really play with. And some are treated with radiation, some drugs, some are patient-derived culture, some are immortalized, some are mouse models. So really nice data compendium. And if you look at if you're looking for clinical data, you have a lot of resources where you can access molecular profiles of tumors. The problem is that a lot of those resources don't really have the phenotype. They don't really tell you this patient has been treated with X, and here's the outcome of the patient in terms of survival or tumor volume kind of reduction, whatnot. So you kind of limit it on the patient side. Surprisingly enough, for immunotherapy, you get tons of those data. I don't really understand why, except maybe that we know immunotherapy is extremely hard, extremely complex. So people kind of felt that sharing that with the community would probably help them find biomarkers. But for chemotherapy and targeted therapy, not so much. So it's great to see so many public pharmacogenomics data sets. So what do you think? They're all standardized, ready to use. They're not standardized, they require a lot of iteration, or is it just impossible? Don't even try. Not really a question. You all know the answer. But the people who said it's impossible, they're not wrong. There are some data, they're just impossible to use because they didn't share the data properly. So you can spend months and months, and virtually there is a missing information. They don't want to share it, you're done. Most of the data set, they're just shared in a way that it's not harmonized, they don't use ontology. So you just spend a lot of time doing the curation. So you're right. So data processing is kind of this dark secret in bioinformatics. Like we spend 60 to 80% of our time wrangling with data. And yet it's one paragraph, if you're lucky, in the paper. So there is this huge technical depth. Like we do a lot of stuff with the data, but we don't really say what because it's boring, right? And everybody's doing something slightly different, and nobody can really compare each other's paper. So what kind of normalization method, what kind of negative controls, what kind of ontologies? So we got in trouble into the lab. Oh yeah, go ahead.
Why are these data sets shared after publication? Is it just like issues with distribution? Is it like hard to host this data?
Yeah. I think we should have a TorBUG seminar on that. So I think there are multiple reasons. The main one, so one is some people don't want to share because they think it's an asset to their own research. Luckily, the new generation of researchers, they're very open-minded. They know, like, I'm doing science, I'm going to share with the community. The other reason is probably the most common one is that it takes a lot of efforts and resources. And usually people do it at the end where the paper is ready, but it's too late. Like you're organizing your data requires a lot of work, and usually from the very beginning. And if you're like me and you have to collaborate with people, you have to be extremely strict about your research data management, because going back a year later and say, oh, by the way, how did you get this data? How did you transform that data? Did you use the right ontology? It's too late. Like people kind of moved on already. So I think there is a bit of a movement, like CHR is requiring a proper research data management strategy. It's going to take years for people to adopt that, but it will come to fruition. But basically, we need money to do this. We need to get the expertise. We need to get the people who know how to do those things. And we need to do it at the beginning of the project. So not an easy thing to do. Michelle is smiling. So we basically in the lab, we run into troubles. Like students were coming to my office and say, oh, I got this result, but the other student got different results on the same data set. Was it really the same data set? So we decided to kind of think about data creation in a different way. And we basically made data creation our own research project. So we developed this platform called Orchestra.ca, where basically you can request a data object. You can say, I want this data set, this version, this date, this pipeline. If this object does not exist, it's going to query the database. It's going to run all the code used to generate that object. It's going to rerun it from scratch. And it's going to generate a data object with a proper DOI. And if the object you request is already existing, you just get the DOI. This is the key. No data is being transformed unless it's scripted. So if there's a typo in the cell line name, a typo in the drug name, or you use a different ontology, it has to be scripted. There is nobody in Excel changing stuff, right? So here you can basically go from the source data, you download it, say, from the Broad Institute, you run that piece of code and you get the object. It's fully transparent. And we make errors more often than not. But with that system, we correct the script and we run it, and we have a new version of the data set. So it's fully transparent. We have a perfect versioning system. So at least when people come to my office, I know with the DOI that they use exactly the same data set. That takes a gigantic amount of work, right? And it's not sexy. You never get it funded, but it's very important to kind of build a foundation for your research. So in terms of modeling, people have been trying to build predictors of, yeah, go ahead.
I'm wondering about,
Yes.
yeah, about database.
Yeah.
We're already assuming that
So
you have to have data.
so when we onboard a data set, we do the curation ourselves, right? So it takes a lot of time, but instead of just saying, hey, here's the data object and forget about the code, we actually say, I don't care about the data object. Give me the code. Somebody else in the lab is rerunning everything from scratch. We actually use kind of a pipeline manager like SnakeMake to make sure that it's fully reproducible. We run it multiple times. When we know that the object is really stable, then we put it on Orchestra. And if we're really kind of passionate about the data set, we can also have multiple ways to process the data. For RNA-seq, do you want Callisto? Do you want Salmon? Whatever. And then we can provide those options. But it's kind of something that's very dynamic. Like if somebody has a new interest, like I have students looking at circular RNA, we're just going to add a circular RNA pipeline within that platform. And now we have a new data object with this new kind of perspective on the data. Now we have the circular RNA, and we know exactly which pipeline, which version of the library has been used. But yeah, it has to be done by the team. If you have your own data set and you give us the script, we can put it on the platform. Easier said than done, but that's the idea, is that the community can contribute their own curation as well. But don't give me your data. Give me a pointer to the raw data and the code. That's all I want to see, because then we are going to recreate the object, right? We need to make sure it's reproducible.
I'm thinking of, like, I have some data that I should pull from a whole bunch of different people's supplementals.
Yeah.
But really online other than.
And I'm sure you had to transform those data to some extent to align them. That's exactly the kind of stuff you can put in Orchestra. So if you send us an email, we can work together to make it happen. And it could be like, you can have the data on astrophysics. I don't care. The system, the concept is run the code. So basically, we don't care so much about the data. We care about the code that generated the object, right? Okay, so tons of papers. You can see it's kind of experimental. And here's a very biased list. This paper is probably the best because it's mine. All the others are really nice too. They are much better. But the cool thing is that some of those predictors, and they use very different machine learning methodology, but those two, they're actually being pushed in clinical setting. Like they created companies that's developing a product that clinicians can use in their trials. So it's very rewarding to see that researchers push the concept so far that now you have a product for the patient. 99.999% of my work is actually not going to end up in the clinic. And it's very, very frustrating. So I really admire people who manage to kind of go through all the regulatory phases and get the clinicians involved. But I don't want to talk too much about the models, those models themselves, but what you have to think about when you build those, because there are a zillion ways to build predictors. And I don't want to go into those things, but I would like to discuss about what we call the hallmarks of building a predictor of drug response. And that's kind of a work with Trey Heidecker at UCSD. It's going to be published in Cancer Discovery very soon. So the first one is data actionability. So think about it. If you want to use like methylation from fresh frozen tissues, you may get super high quality data, but it's going to be very hard to translate into the clinic. Fresh frozen is just hard to implement. And methylation assays, even some methylation is okay, but it's not really kind of a clinical assay that people use very often. But if you do DNA on FFPE, easy peasy, we do that all the time, right? So you basically have to ask yourself, if I really want to translate my predictor into the clinic, I'm probably going to have to make some very hard choice at the beginning about the kind of assay, the kind of samples I'm going to use. And from an ethical perspective, sometimes you don't need to run a new assay in the clinic. Sometimes you can just use existing data, but those data are being shared, especially patient data are very sensitive. They're being shared usually with the identification, some kind of data governance, you have to access data. So there's a lot of ethical consideration regarding the sharing of those patient data. And also you have to ask yourself, is the assay invasive? Is the assay expensive? If it's only gonna benefit rich people, probably see the problem here. If it's invasive, sometimes you cannot even do that. Like if you have a liver cancer, a biopsy could actually spread the cancer cells, so it's actually dangerous. So you have to think about the technology in different ways. And there are big differences between research assays and clinical assays. In research, we always, whine about the lack of money, but the reality is that sometimes we do assays that are 20K for one sample. Right? We do like this single cell spatial transcriptomics one. That's never gonna work really in the clinic. So we basically have very little to no barrier to complexity and cost and samples. And we measure a lot of features, and sometimes they're very noisy because we don't need regulation regarding the clinical approval for the clinical validity of the assay, right? We just run whatever technology we want. In the clinic, it's completely different. It has to be certified. The cost is a big issue, the kind of samples you use. So very different consideration between research and clinics. That's why you have to think about it. What I really like, so in the clinic, you have obviously clinical data and imaging that are being done as part of standard of care. So those data are already there. Sometimes hard to extract, like radiology, we're facing the same issue. Like it's just hard to get into the clinical databases, but the data exists, And sometimes the data don't exist. If you ask for methylation data, it's not one in standard of care. So if you can leverage things that exist, that's great. What I found really, really nice that there are a couple of researchers that do something really stupid and genius at the same time. So for instance, this paper, they did spatial transcriptomics, very, very expensive. And they asked, from a pathological ask, they asked a question, can I predict the level, the gene expression level of every single gene just based on the image of that spot? And of course you can. I mean, that's why we invented spatial transcriptomics, right? But maybe you can get close enough to be able to predict, let's say, the CD8 T cell gene expression signature on that spot. You don't need to be perfect, you just need to be good enough. And suddenly, this very expensive assay is being translated into a pathological assay, which costs you $0. Right? It exists already. So I think that kind of thing is kind of stupid and genius at the same time. You really kind of use research to teach some, to train a surrogate biomarker, a surrogate model on data that exists in the clinic. I kind of really like this way of thinking. So the second hallmark is the architecture. You can have a model that's very simple, a model that's very, okay. A model that's very simple, a model that's very complex. So you have to make some very strict modeling choices. And basically in the lab, we use Occam's razor. Just do as simple as you can, increase complexity and be very careful to make sure that the gain in predictive value is proportional to the complexity you put in the model. And so at the end of the day, if you look at the papers, they're mostly multivariate nonlinear. Why? Because it's fancy, because it's not boring, because you can publish it, right? But is it going to really lead to a clinical biomarker you can use in the clinic? Very, very rarely. So I'm just being sarcastic here, but you really have to choose the right complexity. So in terms of benchmarking, your read papers, they always look great, including mine. When it comes to validate it on a completely new data set that the authors never had access to, they 90% fall apart, right? So you should never, ever trust a paper unless you can generate your own evidence that there is something real there. And so this idea of benchmarking is at the very least within the paper, they should have done a good job at trying to collect multiple independent data sets and try to validate their thing as much as possible. I call it within domain in the sense that if you use cell lines to train a predictor, you can validate it on cell lines. Fine, you don't have to jump directly to patient. And if you do it rigorously, at least you can basically detect the bad models early on, and not going to waste resources on those. Yeah, we can talk about this later on, but this idea of researchers trying until it works and publish, it creates a huge bias. If you have to register your analysis like clinical trials, we will never publish a paper because the first thing you think of never really worked, right? So if you had to stop there, it would be the end of research. But people have done like competitions where they have like this kind of strict way of comparing models, which is really cool because then you don't really control the way you test the model. So you have less bias there. So generalizability is basically trying to use the model with its intended use. So if you want to deploy that in the clinic, you should test it on clinical data that have been collected in the real world and could be retrospective and prospective. So it's not the same thing. Benchmarking is really just, I kind of use the same research data. This is more like, I'm gonna use the data that the model will actually face in real life. And I can tell you, most models will fail that test. And so you also have to document very precisely your model's indication. When does it work? If it only works for subtype of a given cancer, you have to document it. Otherwise, people are just going to use the model in the wrong indication. So that brings me to one of the last hallmarks. It's like, if it only works in Caucasian male, I'm not sure it's a great model. It might be beneficial, but it doesn't sound that fair, right? So a lot of the data sets we're dealing with are extremely biased. Like if you look at the genomics data sets, mostly from European descent. If you look at, so sex is usually a bit biased. If you look at, as I said, preclinical models, a lot of tumors don't grow in the dish. So by definition, there are sometimes that are not represented in your preclinical model. So by definition, you're kind of biased to start with. Your model probably not gonna work for those, but you have to be aware of those biases. And from an ethical perspective, this is extremely important, and nobody's really looking at this from a biomarker perspective. So, but no, I think the awareness is there, and people really start focusing more and more on that. And it's because you have a lot of studies that basically, especially AI, it's all fancy and stuff, but it's very complex, and so AI can detect race in imaging super easily. How do you remove that bias from the model is super, super hard. So here they've tried very hard, and they basically came to the conclusion that whatever they tried, the model can always predict race from X-ray, right? Mm-hmm. So it's kind of, they really don't understand what's going on, right? So many different sources of biases. One that's kind of funny is the measurement bias. And I don't know if you remember the launch of the Apple Watch. It didn't really work on dark skin. So they had to go back, modify the algorithm so that it would work kind of equally between light skin and dark skin. That's kind of the bias you really need to take into account. And then I have two more hallmarks. Am I allowed?
You got 2 minutes.
2 minutes, 1 minute per hallmark. Okay. So everybody dreams of a biomarker, a predictor that's could be interpretable so that you know what the prediction is being made, right? And some are black boxes and some are, you'd really try to make them easy to understand. And obviously from a clinical perspective, if you understand how the predictions are made, like clinicians are more likely to trust it. But I would argue there are probably two scenarios. One is fully interpretable and it's aligned with medical evidence. So it's easy, you go for it, everybody's gonna trust it. If you have a black box, but you have a very strong track record of very good prediction, people will still use it. I mean, people use in the plane, pilot use the autopilot. They don't know exactly how the algorithm works, but it's been working for 50 years. So they kind of trust the model, even so it's a bit of a black box. And the last one, before Nia kills me, it's all about accessibility and reproducibility. And it's a little bit of trust as well. So if you publish, as a researcher, if you publish a predictor, sharing the code, sharing the data, sharing all the documentation about the software environment is very important for people to be able to poke into it and scrutinize, oh, if I change this parameter, if I change the data, if I do this, if I remove a patient, does it still work? Like, you need to let people kind of challenge your research a bit so that they can trust it, but more importantly, they can reuse it in new contexts. And again, it's really increasing trust, but I would argue that it's really increasing return on investment. If you give me $1 million for a project, and I succeed, great. I fail, you lost everything. If you give me $1 million and I share all the data and code, I fail, somebody else may succeed. Or it may be used in another research project, right? So huge difference in return on investment. And it's not new. We have tons of technology to share data, computer code, software environment. So it's not a technical problem. It's an expertise, it's a resources, it's a willingness problem. Oh, and by the way, your university wants to do open science, but they also want you to commercialize. So good luck with that. So yeah, so basically the point here is that a lot of studies don't share anything. And if you think about it, it's not a scientific paper then. It's just a commercial, just an ad for technology that you don't really understand, that you cannot reuse. You know, I was hoping to poke a bit of fun into Google, but you know, we'll do it next time. But they're particularly crafty in not releasing anything. It's pretty amazing. And so we kind of, with this kind of hallmarks paper, we have a bit of a checklist to kind of help you think about those things when you publish. So if once it's publicly available, you can use it for your next paper if you feel like it. And sometimes you can also have fun. Like this is one of my models, and you can criticize. Oh well, I use very good actionable data types, but my generalizability sucks. I didn't even look at fairness. I'm very interpretable. Like you can really start scoring what you read in the literature to understand the pros and cons of a given model. Yeah, we can skip that one. And so basically, my point here is that we need more clinical trial data because it's very important to be able to test the validity of those classifiers. And at the end of the day, if you do this kind of research, never tell clinicians that we're going to replace them. First of all, because it's not true. Second of all, you need their collaboration to do anything anyway. What you want to do is to augment them. It's really to kind of help them making the best decision based on more complex data that they usually can handle on a daily basis. So there is a bit of pitch that needs to happen. Jeff Hinton, even so, is not overpriced, didn't really have one. So we're going to replace all radiologists. Like, it didn't really happen. It's not going to happen anytime soon. Maybe one day, but not in the next 20 years, I suppose. So with that, those are the real people doing the work on the ground. I'm just here for the advertisement. And thank you for the fundraising.